So good morning, everyone. It's my great pleasure to have Dr. Yi Gu join us for the seminar today. So Dr. Gu received her Ph.D. from Johns Hopkins University and finished her postdoctoral training with David Tank at Princeton. During her postdoc, she made a major breakthrough published in Cell, revealing a map-like microorganisms of gray cell in the medial entorhinal cortex, or MEC. To put this in context, the 2014 Nobel Prize was awarded for discovering that MEC and hippocampus form the brain's internal GPS. Dr. Gu's discovery provided one of the very first demonstration of how the brain physically organizes a map for such a complex cognitive variable. In 2019, she started her own lab at National Institute of Neurological Disorder and Stroke. Today, her group sits at the intersection of biology and computation, combining both like two photon images with virtual reality to encode the neural basis of navigation and memory. While the MEC has long been viewed as a purely spatial module, Dr. Gu's lab is pioneering in showing that the MEC also encodes non-spatial information, integrating sensory and visual landmark details into the internal map. Her lab's most recent work in Nature Communication reveals that the Q cells in the MEC encode not just where a landmark is, but exactly what it is. So let's welcome Dr. Igu for her seminar.
Hi, everyone. My name is Igor. Thank you so much for the great introduction. Yeah, so my lab has mainly two focus. One is the introduction mentioned, we study how the MEC can encode the spatial and non-spatial information. Another main focus is about how the MEC can support spatial memory. So today I will be focusing on the second focus and introducing a story about how slow synaptic plasticity from the hippocampus underlying gradual mapping and fragmentation of novel space by gray cells. So the spatial memory in mammals is highly relying on the hippocampal and entorhinal circuits, which localize in the caudal region of the rodents. And sorry, can people see? Yes, localized at the caudal region of the rodent brain and the medial temporal lobe in the human brain. So when the animal is navigating in an open environment, the hippocampal plate cell fire at a single or a few locations, whereas the entorhinal grid cell fire at multiple locations, tiling a triangular lattice in the open environment. So therefore, this circuit is hypothesized to form a cognitive map of space to guide spatial navigation. And this circuit is very important for spatial memory. A very famous example is a human patient, HM, who lost medial temporal lobe bilaterally and was suffering difficulty in forming novel environmental memories during navigation. And the dysfunction of these circuits is also highly associated with the spatial memory deficits in many disorders such as Alzheimer's disease, temporal lobe epilepsy, and schizophrenia. So my talk today will primarily focus on the medial entorhinal cortex in rodents, and we want to discuss how does gray cell activity in the mouse MEC evolve during spatial learning, and how is this activity associated with the spatial memory. So imagine that you have to learn to navigate an environment with multiple landmarks and complex structures. And you can imagine that the mapping of this environment requires a gradual reconciliation between the actual position and the estimated position based on your cognitive map, so that the error of your internal representation could be gradually corrected by the salient features throughout the navigation process, so that you can have a more correct map for this space. And to understand how the gray cell is playing a role in this process, the ideal technique would be following the gray cell activity during this reconciliation process, so that we can understand how the gray cell activity evolved during the spatial learning process. And to reach this goal, two-photon calcium cellular resolution two-photon imaging would be ideal to track this process across many days of spatial learning. However, most of the previous studies rely on the electrophysiological approach, which suffer the limitation to reliable track large number of neurons during spatial learning process. So despite of this, many of the previous studies show some interesting findings about spatial involvement of gray cell activity during learning. And several studies suggest that the gray cell activity in the novel environment can evolve in an experience-dependent manner. So in this study, there are around 10 gray cells were consistently tracked across 5 to 7 days when the animals was exposed to a novel environment. The authors find that from the switch, during the switch from familiar to normal environment, the gray cells show enlarged spacing, which is the distance between adjacent gray field. And this enlargement of spacing can gradually decrease to a normal level after 7 days of experience. And in another environment, the gray cell population was tracked when when rats were navigating in 2 connected rooms. So the rooms had a boundary, had an invisible boundary, and the rats was freely moving between these rooms. And in the initial exposure, the gray cells in the rats were separately representing these 2 rooms as an independent map. But after around 20 days of travel between these rooms, and the gray cells gradually develop a global mapping across both rooms, suggesting that the gray cells can form global representation in an experience-dependent manner. However, a recent study suggests that the gray cell activity in a novel environment is experience independent. So this study was conducted in virtual reality, and hundreds of gray cells was tracked within 1 day. So this track contains multiple blocks of virtual linear track where the water reward was localized at the same location, but the landmarks were shifted differently. And as you can see that the mice could quickly learn this task by conducting predictive licking before the reward location. However, if looking at the gray cells, and you can see that between those 3 different blocks, the gray cells could quickly switch their pattern and form a stabilized map. So that this study is suggesting that activity of gray cells can form in one-shot exposure to the environment and does not require plasticity of gray cells. So given this conflict across different studies, my lab decided to conduct a better approach to longitudinally track gray cells across multiple days of spatial learning and to investigate how their activity actually evolved during this process. So today's talk, I will be introducing 3 components. So the first part, I will talk about the long-term calcium dynamics of gray cells during spatial learning conducted by my lab. And the second component will talk about our collaboration with Elafis lab on theoretical modeling for a Hebbian plasticity-based mechanism that can explain the gray cell dynamics we observed. And the last, I'm going to introduce a collaboration with another group to test this plasticity using ex vivo physiology. So the first part of long-term imaging was conducted by talented postdoc Dr. Lu Jiachen, Dr. Taylor Malone, and the postdoc Farid Shahid. So to conduct a longitudinal tracking of gray cell activity during spatial learning, we performed 2-photon calcium imaging of mouse MEC by surgically implanting a microprism that could redirect the optical path 90 degrees to reach the superficial of MEC. So here you're looking at the expression of MEC neurons in layer 2 expressing GCAM6. So this technique allowed us to monitor the neurodynamics of MEC in real time while mouse was navigating in virtual reality and under head-fixed condition. So this technique together allow us to track neurodynamics of the same MEC cells across multiple days during spatial learning. So in our typical spatial learning task, the mouse was head fixed under the microscope, and they were exposed to virtual linear tracks containing landmarks and water reward. And water-restricted mice unidirectionally navigating in this track for multiple laps through a teleportation at the end. And in the typical task, we had the mouse first trained in an environment until familiarization, we call them familiar environment. And then we exposed the mice to a normal environment where the new landmarks and the reward were shifted to different locations. And our two-photon imaging was conducted from the last day of a familiar environment across 10 days of spatial learning to investigate the development of calcium activity. So to evaluate how the spatial learning went, we used the reward predictive behavior. So here I'm using the gray color showing you the running speed of a mouse, and the red dots are indicating the licking. And you can see that when the mouse was first exposed to a normal environment, they show uniform running speed and random licking throughout the track. However, after 10 days, the mouse developed this robust reward predictive slowing and licking, suggesting the establishment of their spatial memory to the environment. And our mice exhibited a great variety of behaviors in such tasks. And based on their predictive behavior, we separate them into good and poor performers for the convenience of comparison. So the good performers show high reward predictive licking and slowing in familiar environment. The behavior dropped in novel environment and quickly established to reach the high level during learning, whereas the poor performer consistently showing low predictive behavior throughout the learning. So these two group of mice correspond to successful and unsuccessful learning, so that we could investigate the gray cell dynamics underlying these different conditions. So here I'm showing you the activity of a gray cell, a single gray cell, when the mouse was first exposed to a normal environment. So in one-dimensional linear track, the gray cells could fire at multiple locations and form this multiple gray field on a run-by-run basis. And interestingly, we find that when the animal was navigating in forward direction, those gray fields shifted in backward direction, suggesting that the gray cell activity could change with experience within a day. And we further connected the gray cell activity, the activity of the same gray cells across multiple days, and we found that those spatial fields also shifted across days. And such backward shifted gray fields was less observed in non-gray cells. So to quantify this observation, we classified the gray cell field based on the slope on a run-by-run basis, so that the gray field could be separated into backward field, forward field, and the stationary field, which do not exhibit significant shift. And we found that the percentage of backward shift in gray cells is significantly higher above chance, where the percentage of the other populations were lower than chance. And this is consistent with our observation. And we also found that the percentage of fields that show backward shift is higher in gray cells than non-gray cells. And this percentage was also higher in good performers than the poor performers, suggesting that this field could be related to spatial learning. And we further calculated the number of days the field could last and plotted the duration of this field in this histogram. And we observed that in comparison to the poor performers, the good performers show greater percentage of fields that lasted for longer, for more days. So together, this result indicate that the backward fields in good performers are more abundant and long-lasting, suggesting that these fields could be associated with a spatial memory. And you can imagine that if the field is contributing to the memory, they should form a gradually stabilized dynamics to encode better memory during learning, and this is indeed what we observed. So here is the dynamics of a single backward shift gray fields across days, and you can see that the field started from backward drifting and became gradually stabilized in a more vertical direction. So to quantify these dynamics, we separate this cross-day field into individual day fields, and we quantified the variation of run-by-run field within a day. And we found that indeed the backward shifted gray fields became gradually stabilized, reducing the variation across days. And in contrast, the non-backward fields containing the stationary and forward gray fields did not show such robust stabilization. And the stabilization was also more robust in the good performers than the poor performers. So next, we ask whether this field could be somehow associated with the salient environment features, which is a very important properties to distinguish different environments. So for this analysis, we grouped the grid field between adjacent landmarks, and we call the landmarks in the backward direction as the back landmarks, and the landmarks at the forward running direction as front landmarks. And we summarize the dynamics of grid fields. So here I'm just showing an example of two fields between these landmarks. And you can see that the grid field that is closer to the back landmark show greater stabilization compared to the fields that are further away from the back landmark. And our quantification of grid field stabilization further demonstrates this observation that the grid fields near the back landmark show greater stabilization. We also observed that the grid field between adjacent landmarks, while they initially were distributed uniformly between the landmarks, after spatial learning, the field distributed towards the back landmarks, and the field that passing the back landmark is percentage is relatively lower compared to regular Gaussian distribution. This is suggesting that the back landmark is not only stabilizing the fields, but also preventing the field from passing, so that the grid fields can eventually reach a state that is fragmented by those individual landmarks. So while the above observation is showing the gradual stabilization of the grid field near the landmarks, and now next we ask about their offline dynamics when the animal was not experiencing virtual reality. So as you can imagine, the animals spend most of the time in their cages, and we predict that if there was a fast memory consolidation or predictive plasticity, you can imagine that the backward shift of a grid field that the animal acquired online during virtual reality experience could further shift when they were kept offline in their home cage. And in contrast, if there is a memory loss, we would expect the regression of the field, meaning that the backward shift of the grid field on the next session, on the first run in the next session, would be further in the forward direction compared to the shift in the previous day, meaning that the animal start to lose the learned spatial location advance in the offline period. And if the memory indeed got lost during the offline period, but still accompanied with some memory consolidation, we would expect a partial regression, meaning that the total amount of learned backward shift in the prior day was only partially reset on the next day. Whereas if the whole memory was completely lost without any consolidation, you would expect that the backward memory regression on the next day was beyond the amount of the shift that they learned in the previous day. So to test this hypothesis and see whether there is indeed a consolidation process, we monitor the grid cell activity between adjacent days. Here I'm showing you activity example. And the grid cell show backward shift on a run-by-run basis as their trajectory was reflected here. And you can see that this field indeed show regression relative to each other, suggesting that the grid fields, this regression was quantified here by a greater portion of regression events compared to the progression events. This is suggesting that there is indeed a memory loss when the animal was kept offline in their home cage. And then we further asked whether there is indeed consolidation in this process by measuring the amount of total shift in the previous day compared to the amount of regression on the next day. And we found that indeed most of the regression were partial, suggesting that there is a consolidation process, but it's very slow. It requires some online experience and a longer time scale to consolidate than the plasticity induction. So together, this result is suggesting that the dynamics of individual grid cells became backward shifted with experience, but gradually consolidated into a more stable map during learning. So our next analysis is focused on the population activities of grid cells, not only limited to the backward shifting fields. So before we talk about that, I would like to introduce a concept about how the grid fields was formed in one-dimensional linear track. As I introduced previously, the classical grid cell field was measured as a two-dimensional hexagonal lattice in open environment. And our recent theoretical study showing that the gray cell activity in a one-dimensional linear track could be approximated as a slice through a two-dimensional lattice. So every time when this linear track slices through the activity bump in this two-dimensional lattice, it will evoke a corresponding fields in the one-dimensional linear track. And since the gray cells form continuous attractor network, so multiple gray cells should form a coherent activity showing as parallel lattices in this two-dimensional manifold. And therefore, their population activity can stay low dimensional when the animals is traveling in this one-dimensional linear track. So, however, as I show you that the gray cell backward shifted, some gray cell field shift backward, some field shift forward, and they also shift differently in relation to the landmark locations. And the next question we ask is whether during spatial learning, gray cells still maintain their coherent activity, and if they do, how does their activity as a population evolve in this low-dimensional manifold? So as you can imagine, if a single gray cell activity is triggered by the slice through a two-dimensional lattice, and if the lattice was shifted during spatial learning, you can expect that the gray fields should all of this gray cell along the track should all shifted coherently in the same manner. However, we found that as we observe the activity of this single gray cell at multiple locations along the track, some of the field could shift backward, some of the field will show stationary dynamics or shift differently. So this result is suggesting that the trajectory along the two-dimensional manifold is distorted, not limited to a straight linear track. So, and we expect that if the gray cells still exhibit coherent activity together, the gray cell, their activity trajectory should be distorted together in this manifold. And indeed, this is what we found. So here, I'm showing you the activity of multiple gray cells labeled by different colors, and their field could occur around similar locations, but you find that those fields across different gray cells all shifted together. And we also quantify this observation by calculating the correlation between the field trajectory as a function of their distance along the track. We find that the nearby gray fields across different cells could shift more closely compared to the gray fields that are further away. So together, this result suggesting that the gray field in the spatial learning still maintain coherent activity, and they show distorted trajectory, but the trajectory were distorted together as a population, so that the population can still maintain on a two-dimensional manifold. You have a question?
Is this trajectory like a distortion, like accompanied with some actual, like maybe animals' behavior, like animals after maybe thinking, like they change their trajectory? That is changing this slice on this neural sheet.
Yeah, we'll talk about that later. So yeah, thanks. So based on this observation, we computed the population trajectory of the gray cell, multiple gray cells, using their activity, and those together their activity indeed form a coherent activity bump moving along the dimensions in this unit rhombus, which is the basic unit of this two-dimensional lattice. And for the convenience of visualization, we projected this trajectory in this vertical direction. And here I'm showing you the trajectory of gray cell population on one day across the track during spatial learning. And we plotted this trajectory of the same gray cell population across multiple days during learning. And then we asked how their activity this activity pattern developed. So to answer this question, we calculated 3 parameters. The first parameter is the distance between the trajectory, which is the distance between the corresponding track locations along the trajectory on different days. And we also calculate the start location of this trajectory, and which is the distance between those 2 start points, the start point of adjacent trajectory within unit rhombus. We also calculated how the trajectory rotated during learning. So based on all these 3 measurements, we find that the trajectory distance, the lateral shift of their starting point, and the rotations altogether show gradual decreased change, suggesting that during spatial learning, the gray cell population activity also form gradually stabilized map. So next, I would like to address the distortion question that I mentioned about. So since the gray cells show different spatial shift with their different distances around the landmark, we hypothesized that the landmark could distort this gray cell trajectory. So to test this hypothesis, we quantified the population trajectory jump as along the track location. As you can imagine, when the population activity was not jumping, was not distorted, the population activity jump should form uniformed jump in every unit of centimeters along the track. However, if there was a distortion, there should be a greater jump around the salient features, for example, landmarks. And indeed, we observed that here when we were plotting the activity trajectory jump along the track and compared the jump within the landmarks and between landmarks, and we found that the landmark indeed correspond to a greater activity jump compared to the out landmark locations. And in addition to the landmark, we also found that another location that is near the reward also caused a greater activity jump of the trajectory. So to specifically isolate the effect of the landmarks, here we remove the reward location and quantify the jump within the landmarks versus between the landmarks. And we indeed see that there is a greater jump between the landmark locations, suggesting the distortion effect by the landmark. Now we further asked how this distortion changed during spatial learning by quantifying the influence of the landmark to take the difference between the landmark jump within and between the landmark locations. And we found that this distortion in that this influence by the landmark increased during spatial learning, suggesting that there is an increased landmark influence to this trajectory during learning. So that overall, this activity dynamic suggesting that the gray cell population can be distorted by the landmarks, and this distortion gradually increase. So overall, this above population dynamic is suggesting that during spatial learning, the gray cell population activity could shift and rotate and became increasingly distorted by these landmarks. And gradually, this population activity can reach a stabilized dynamics and became fragmented by the landmarks. And we hypothesized that the shift and the rotation could underline the backward shift of the gray cell fields in this process. Then to better understand this mechanism, we collaborated with Dr. Ilafis' lab at MIT to conduct a theoretical modeling to explain our observation. So before going to the modeling, I would first like to introduce the circuits in the between hippocampus and MEC. So the gray cells we imaged are localized in the superficial layer of MEC, localized in the layer 2 of the MEC. And the gray cells localized here and the plate cells are localized in the hippocampus could form reciprocal connections between the MEC. And mostly the CE1 subiculum could form direct and indirect inputs to the superficial and MEC of the MEC, and the indirect inputs was relayed through MEC layer 5. And in the meantime, the entorhinal cortex could also receive sensory information and send the projection back to the hippocampus. So our model was constructed in the way that is based on the previous published vector hash model from Ilafis' lab. So the model hypothesized that when animal was exposed to a normal environment, the sensory information quickly come in and induce a change in the hippocampal activity. And the hippocampal activity further drive the plasticity of gray cells in a time-delayed manner so that they can shift the gray fields in specific manner. And this, particularly, this plasticity is temporarily delayed, Hebbian-like plasticity, and combined with a slow consolidation so that the gray cell could shift and also became gradually stabilized by a consolidation process we observed previously. So in particular, the hippocampal inputs arrive at the gray cell synapses in a time-delayed manner before they potentiate those synapses. And this time delay is causing the backward shift of gray cell fields during spatial learning. So here I'm showing you an output of this model. As you can see that the modeled grid fields could gradually stabilize, a backward shift and gradually stabilize near the landmarks. And this observation is recapitulating with our experimental observation. And to further understand the mechanism, we generate the model in different conditions. So in the top, the model was what I observed, what I introduced previously, was the plasticity with a delay, and the middle model was a condition where the plasticity was completely removed from the network. In the last condition, the plasticity from gray cells, from hippocampus to gray cells, was set without a temporal delay. As you can see that while the temporal delay caused backward shift in the gray cells and no plasticity condition, the gray fields was noisy and did not show directional fields. And the plasticity without delay, the gray cells quickly shift but can form a very quickly stabilized spatial map. So therefore, this result is suggesting that the delayed plasticity can drive more backward fields shift in the gray cells. And also we calculated the spatial decoding error in these three conditions. And we found that the synaptic plasticity is required for stabilization, as the no plasticity condition does not reach improved spatial decoding error during learning. And if we further calculated the fragmentation of the grid fields by the spatial landmarks, and we found that the increased influence index from the landmarks is only specifically increased, observed in the plasticity with the delay condition, but not in other conditions, indicating that the temporally delayed plasticity specifically shapes the landmark fragmented map during spatial learning. So together, this result is suggesting that the temporal delayed synaptic plasticity from the hippocampus to the gray cells, plus slow consolidation, is potentially responsible for their backward shift and gradual stabilization, so that this grid map could gradually reconcile between external landmarks and the internal representation to achieve a precise spatial representation. So next, we would like to investigate the possibility of this plasticity using the experimental approach. We collaborated with Veronica Alvarez's lab at NIMH to conduct ex vivo recording slice physiology to explore the nature of this plasticity. So as I mentioned previously, in comparison to the when we compare good and poor performers, and we found that there was a greater proportion of fields that are backward shifted, and also they undergo more robust stabilization during spatial learning. And we hypothesized that the network state of good performers favors LTP more than that of poor performers, so that the good performer could continuously learn multiple environments. To test this hypothesis, we trained the animal through a novel environment, as mentioned previously, and we separate them into good and poor performers, and as we did previously. And we prepared ex vivo slices containing hippocampal circuit and also the MEC. So we tested the long-term potentiation by stimulating the hippocampal input and recorded the superfield potential in superficial layer of the MEC. And interestingly, we find that upon high-frequency stimulation, the good performer indeed exhibit long-term potentiation-like response, where this response was not observed in the poor performers. So this result is suggesting that the good and poor performers exhibit different network dynamics after spatial learning. So to better understand this underlying mechanism, so we specifically express the channel rhodopsin in the hippocampus to isolate the inputs from the hippocampus and recorded the response in the MEC upon optogenetic stimulation. And similarly, we separate the trained animals into good and poor performers and optogenetically stimulate the fibers in layer 5 of the MEC and performed patch clamp recording of individual individual neurons in superficial layer of MEC. We preferentially target stellate cells, which is more likely to contain the gray cells observed experimentally. So here's our observation. So our whole cell recording is the first finding is we found that the good performer, the LEC neurons in the superficial layer show higher EI ratio, which is a balance between excitation and inhibition of individual cells. And we also found that this EI ratio is positively associated with spatial learning performance, suggesting their experience-dependent relationship. And we further asked what kind of input is driving this differential EI ratio between good and poor performers by separately analyzing the EPSC and IPSC. And we found that in the good performers, they had reduced the EPSC as well as reduced IPSC, but the extent of reduction was greater in the IPSC, and this IPSC amplitude was closely associated with spatial learning behavior. So this result is suggesting that when the animal is more is well performed during spatial learning tasks, they show higher EI ratio, which is preferentially contributed by the reduction of inhibition that they received. And to further understand the underlying mechanism between the communication between hippocampus and the entorhinal cortex, we analyzed the latency of the EPSC based on the previous knowledge that the hippocampus could generate direct and indirect inputs to superficial layer of the MEC neurons. So the direct inputs coming through the CE1 subiculum, they could recruit the local interneurons and also direct project to the excitatory neurons. And this pathway is predominantly driving strong inhibition to the superficial MEC. And on the other hand, the indirect input was relayed through MEC layer 5, again, also recruiting local interneurons and exciting the excitatory neurons. So to determine the contribution of these direct and indirect inputs, we investigated the optical EPSC and IPSC latency in the good and poor performers. And we found that, interestingly, the good performers is missing the components of short latency EPSC and as well as IPSC, suggesting that there is less EPSC and IPSC inputs directly coming from the short direct pathway. We further investigated whether this EPSC and IPSC latency is associated with behavior, and the EI ratio we observed previously. We found that indeed the median EPSC, the minimal optical IPSC latency is positively associated with spatial learning performance, but not EPSC. And this IPSC latency is also associated with the EI ratio we previously observed. So this result together is suggesting that the good performance corresponds to relatively weakened direct inputs, which largely convey inhibitory inputs to this network. And this could lead to higher EI ratio under the good performance. So together, this result is suggesting that while the hippocampal entorhinal interactions can form in the way that the hippocampal have direct and indirect inputs to the and so superficial MEC, in the good performers, there is a relatively weakened direct inputs leading to weaker inhibition. And this is creating a condition that favors the synaptic plasticity, the long-term potentiation driven by the indirect inputs. And since this potentiation is potentially driven by the indirect inputs, the hippocampal inputs arrives at the superficial MEC in a time-delayed manner, and this could cause this temporal delayed plasticity and then leads to the backward shift into grid fields. This system overall together enable the reconciliation and precise spatial encoding of the gray cell system during spatial learning. And our model also suggests that the gray cell activity produce fixed inputs to the hippocampus, which could potentially drive the backward shifted place cell fields that people observed previously during. So with this, I would like to thank my lab members who significantly contributed this work, and also the collaborators at MIT and NMH for their collaborative work. And I would also like to thank other people in my lab and the funding resource. Thank you very much for your attention. I would like to take questions. Yes.
Oh, that was great. I was really curious about the increasing distortion, like around the landmarks and the rewards with learning. I mean, that seems like, it must be really important, right? And I'm just curious, why you think that's happening. Is it something about, optimizing the representation of the stimuli? And somehow that representation is combined with the spatial representation that's causing the distortion, or how do you think about that?
So the distortion is very important because it seems to be highlighting the salient features. But what we observed seemed to be conflict with another the early study that I introduced when the animal was navigating in a joint environment, and the map initially was fragmented and become globally coherent. So we think that the difference here is mostly because our linear track was very long. It's like 10-meter track. Because there's a complexity of this track, the animal was probably was unable to see a distal cue to signal them is a coherent track. They had to break up their map to fragment these individual pieces in order to map the whole environment. I think, and because the animal was navigating unidirectionally rather than bidirectionally, and without a distal cue, it could be difficult, and this could be a more efficient strategy for them to map the environment. I'm not sure whether I answer your question, but in this other previous condition, the animal was able to run back and forth. It's a relatively simple environment, so that it could gradually form a coherent map.
Yeah. It also doesn't look like there's individual salient spatial cues in that environment unless there's.
There wasn't individual cues in that environment. It was a very simple environment. Yeah.
Thanks. For the time-delayed Hebbian feedback, I understand that certainly if the learning was quick, you weren't able to explain the data in the sense of this gradual backwards and then stationary. So the time delay was important to capture the data.
Right.
What does the time delay buy you? Why would the system want to do that? Is it getting the landmarks more accurately encoded? Why delay things?
So yeah, this is a great question. So the time delay, we actually have modeled many other conditions where, like, for example, if there was no time delay, the network could quickly stabilize. But we found that, yes, this condition does not highlight the landmark locations. We think the backward delay could enable the system to better represent the salient feature of the environment. But in terms of the stabilization, indeed, the no delayed condition could lead to faster stabilization, but it wouldn't form
Unless it's in the wrong spot.
It's where it is. Yeah, so it wouldn't be more environment specific in this sense. Yeah.
So grid cells, they have a theta sequence and like Moses shows, but would it be possible instead of the grid coding per se has changed, maybe how they sweep this environment has changed over time, how they get used to the environment, and maybe they sweep longer, you may appear this grid coding earlier, or how the direction of a sweep changed over time, then that creates a distortion in the environment, let's say grid coding.
Yeah, that's a good thinking. But I think our calcium imaging doesn't have the resolution to record the sweeping, but I think that could be something that we can integrate in the model to better explain the dynamics. Yeah, I think that's a very good idea.
We have a question online from Michelle Miller saying, given the gray cell, the place fields are also observed to shift in CA1. How does the shift occurring in early presynaptic region impact the stability of the gray cell? Does the gray cell's stability rely on plasticity plus delay and stable hippocampal place cells?
So beyond the 3 different forms of model we show, we actually modeled many other conditions where the gray cell, so in this model we show that there is Hebbian plasticity from sensory to hippocampal. There is like faster learning from sensory to hippocampal neurons, and then slow learning from hippocampal to gray cells. And we show that this slow synaptic plasticity is important to stabilize the gray fields, and the gray cell can send fixed inputs to the hippocampus, meaning that there is no plasticity from the plate cell from gray cells to the plate cell side. So that our model showed that the backward shift in the plate cell and the gray cells were coherent, and then they stabilize gradually together during spatial learning. And also, this sensory input is important for forming this landmark anchored and stabilized grid fields and there was no plasticity in this condition, the whole network wouldn't be stabilized. Yeah.
More questions?
Could you possibly comment on how the synaptic plasticity would look different for subjects that have like memory disorders? So how would would that get disrupted, like the stabilization?
In disease condition, right? We do have a study focusing on the tau pathology condition, like we had like PS19 model, and we compared the backward shifting fields, and the PS19 model indeed have more disrupted fields and relatively less directional fields compared to the wild type condition.
So that backward shift looks, how does that look like for?
So there is a lower percentage of backward shift fields and also forward, like the field basically became messier, they were less directional. And if you look at the run by run variation, that was lower in those PS19 models compared to the wild type condition.
So I have one more question. So how does this one-dimensional slicing in the grid neural sheet distortion can be expanded to interpret this 2D especially this 2D data this grid spacing shrinks. So it's
the shrink of the grid.
Because to translate that, you need to think of very complicated way of distortion how do you think about translating into the 2D findings?
Right, so that's a good question. So the backward shift phenomenon was not reported in 2D, but there was one paper from Lisa Giacomo's lab. They reported when the animal was running in an open environment, the gray cell tend to be more stabilized near the boundaries than they are further away into the center of the track. So there is a potential link between the landmark boundary effect to the gray fields, even in two-dimensional environment. But indeed, people have not observed the backward shift in the gray fields. I think if we were able to analyze the data, maybe we could find some hint of that. And in terms of how this is associated with the spatial spacing change, in 1D activity, we also observed the increased spacing upon the first day in normal environment and gradually decrease. But in our data, we found is mostly triggered by the fact that the gray fields were not very stable. They were hardly identified as a field, and leading to apparently larger spacing between the gray cells because there are fewer fields. So so I think it's still need more work to how to put this together, but this is our observation so far. Yeah.
Thank you.
Let's thank Dr. Gu again for